Okay, so I'm going to be talking to you about the human liver fibrosis model we are developing at STAMI. This is also one of the numerous projects in PARC. And I will also be explaining you a little bit what do I mean by new approach methodologies and how this is relevant in the context of chemical risk assessment. So liver fibrosis occurs when the normal tissue of the liver becomes replaced by scar tissue. And this fibrotic tissue deposition occurs on a spectrum of degrees of severity, being cirrhosis, a more advanced and prolonged stage of liver fibrosis that in some occasions can lead to hepatocellular carcinoma. And we know from the literature that certain occupational chemicals are associated with liver diseases. So for example, carbon tetrachloride, which is a precursor of refrigerants and used as a cleaning agent, and allyl alcohol, which is a solvent and a herbicide, have been associated with virtually all the spectrum of acute and chronic liver diseases. And vinyl chloride is the monomer for making polyvinyl chloride plastics, the very well-known PVC plastics. And this has been associated with an excess on the incidences of all these stages of liver fibrosis in workers that were exposed to the vapors of vinyl chloride. However, there is a large gap in epidemiological data on occupational liver diseases. This is rather a critical knowledge gap. When we see, so on the bottom of this picture, we have around 200,000 chemicals that are registered in the two major regulations, chemical regulations in Europe, namely REACH and CLP. And on the top, we see those chemicals, around 700, for which we have occupational exposure levels or limits. And this means that those chemicals up there, they are extensively characterized. We have a lot of toxicological information, but this is just the tip of the iceberg. And we know that under the surface, and as reported in the CAS registry, there is around 200 million chemicals for which we lack any toxicological information. So this becomes, so the lack of epidemiological data on occupational liver diseases becomes very relevant in light of all the vast and unknown chemical universe. And this is how chemical risk assessment has been performed in the past by using animal models. And although animal models have served an important function, they also have a series of disadvantages. So they are highly time and resource consuming. They cannot faithfully replicate human physiology. They do not cover all health adverse outcomes. And we have been exposing animals to one substance, but we know that in the real life we are exposed to mixtures of substances. And last but not least, the strong ethical considerations attached to animal experimentation. So this is where PARC comes into place, because one of the main objectives of PARC is to transition from this old or classical way of doing chemical risk assessment into the new chemical risk assessment, or so-called next generation risk assessment. And this next generation risk assessment builds upon a relatively new buzzword in the toxicology field, which is NAMs, new approach methodologies. And these methodologies do not rely on animals, and they can be grouped into in silico methods and techniques, which consist of computational and modeling methods, in vitro techniques, experimental techniques in the lab, so cell cultures and more advanced cell cultures such as tissue and organ engineering, high throughput screening and imaging, and we will also have all the collection of the omic techniques. And again, an important aspect of NAMs is that they are based on human data. So these cells are human cells. This computational and modeling uses human data. So together with our external PARC partners, we will be bringing different NAMs or techniques from the different institutions targeting liver fibrosis. So we will have some more simple monolayer cultures to more advanced cell cultures such as liver spheroids and the multicellular transwell culture, which is the model we are developing in STAMI. And we will also be using some computational methods such as text mining, where we will extract data from the literature in order to make gene networks that are relevant for liver fibrosis. And we will also be using transcriptomic analysis to further gain insight into hepatotoxic substances. So looking a bit more into the model we are developing, we will be trying to mimic the liver, the human liver. So for this, we will have the 4 main cell types involved in liver fibrosis, which is hepatocytes, which is the cell that carries the main functions of the liver. Hepatic stellate cells, which is a cell that gives support and also plays additional roles in the liver. Liver sinusoidal endothelial cells, which are the cells that line the blood vessels that penetrate the liver, and macrophages, which is a type of immune cell. And these 4 cell types, they will be organized into 2 compartments that are separated by a porous membrane. So mimicking, in the upper compartment, we will be mimicking the liver parenchyma that consists of hepatocytes and stellate cells, and the lower compartment we will be mimicking those blood vessels that come into the liver. And we started doing some initial characterization. We have these 2 cells in the lab, hepatocytes and stellate cells, and the upper picture we see the typical aggregate and polygonal morphology of hepatocytes, and on the lower picture we see also the characteristic elongated and star-shaped morphology of the stellate cells. And we also did some functional characterization. So we see that the expression of albumin, which is a protein that is produced and secreted by hepatocytes, and the expression of smooth muscle actin, which is a specific marker for the active hepatic stellate cells. Okay, so once we have all the 4 cell types together growing in our cellular model, we will then expose our cell cultures into 3 preselected hepatotoxicants. We selected those because we know that at least in animal models, they trigger liver fibrosis. So this is an important control to check whether these substances also will trigger the hallmarks of liver fibrosis in our model. And we will expose at defined concentrations and defined time points. And we will be able to identify and characterize the molecular hallmarks of liver fibrosis. So this will be, first we will be able to measure injury to hepatocytes, and then we will be able to measure the macrophage infiltration, because we have the macrophages in this bottom compartment, and because we have this porous membrane, we hope that they migrate into the upper compartment, and then we can measure them together with hepatocytes and stellate cells. Then we can also measure the secretion of inflammatory mediators, and at later stages of the development of liver fibrosis, we will measure the activation of stellate cells, and it's actually the stellate cells, the ones that produce the scar tissue. So this is a relatively important event, and at the end, we will also be able to see this scar tissue, which is the extracellular matrix deposition. And so with this, we hope as long-term social impacts or expectations to contribute to reduce the use of animal experimentation, to increase the throughput of chemical substance tests, and reduce the exposure to hepatotoxic substances at work, and overall contribute to the improvement and well-being and of health and well-being of workers. And this is just a final slide of all the contributing institutions in PARC. So, yeah, thank you for your attention.